Welcome to this class on Neuroscience of Human Movement. This is part 6 of the discussion on primary motor cortex. So, in today's class the activity of a population of neurons in M1 in primary motor cortex. So, in the previous class or in one of the previous classes we saw a population of neurons can predict the direction in which the monkey is trying to move. So, if I am recording from a whole bunch of neurons, depending on their population activity, depending on the overall activity of all these neurons, I can predict what is the direction in which this monkey is going to move. As in, each neuron has a directional tuning curve or a preferred direction. Summing all the activities of all these neurons at a given point in time will give you an approximate prediction of the direction in which the movement is going to happen. This is the work of Postulus that we discussed in the previous class. In today's class we will discuss something else. In today's class we will discuss the instantaneous trajectories being predicted by populations of neurons. So, we will discuss that. In the second topic we will be discussing how the activity of M1 neurons varies depending on the forces required to maintain the direction against external loads. So, in this case, in the previous case, what is the difference between the previous case and this case? In this case the monkey's movement is in a particular direction. Let us suppose the monkey is moving in that direction. It is possible that there is no load that is acting, that would basically take you to the Georgopoulos experiment. Let us remind ourselves what this experiment is. This is the center out reaching task. So, there is a center and there are multiple targets, okay. These are the targets. These are the various targets, is it not? So, if the monkey is reaching to that point or to that point or to that point, for example, it could reach to any of these 8 targets. Now, if we are recording just the monkey's movement when there is no external load, that is basically Georgopoulos experiment. If I add an external load that either partially or completely opposes the movement, suppose the monkey is trying to make the movement toward the right like that or like that, and the external load is acting in the opposite direction, say, what will happen? That means you would expect that for the movement to happen there will be a greater activity of these neurons. Is that happening? So, that is the second question, okay. And the third question is about what happens when there is no real movement, but force is produced against a non-moving object or isometric force production, okay. So, these are the 3 things we want to discuss in today's class. So, Andy Schwartz and his colleagues, they studied the activity of about 240 M1 neurons. So, this is nearly, I think, nearly 240 neurons in the primary motor cortex of live behaving monkeys. So, the movement trajectories that were required to be made were spirals. So, this is movement and movement. This movement can be this spiral can be made from the outside to the inside as it is happening in A, or from the inside to the outside as it is happening in B. In other words, I could either do that is I am going from outside to the inside. In this case the monkey is going from inside to the outside, is it not? So, there are 2 possibilities, okay. These so figures A and B represent the actual movement trajectories, okay. From these movement trajectories it is possible for me to decompose this into directions, instantaneous directions and velocities. So, when I do that I get a time series or a temporal sequence of vectors. So, this is that temporal sequence, so that is time. So, I am going to get a time series of vectors with the instantaneous directions and speeds encoded in them. So, and that is for the movement A and this is for the movement B. again this is suppose this is population activity. Okay, this is for the movement A and this is for the movement B. So, that is for that one. Okay. This is the decomposition of the actual trajectory into the instantaneous directions and velocity vectors. Now, from 240 neurons it is possible for me to get directional tuning curves like in the case of Georgopoulos, and that is found to be this approximately. So, I am recording from these neurons in simultaneously while movement is also being recorded, is it not? So, with these neurons, their activity, the instantaneous directions and the level of activity can be represented as vectors like in the case of Georgopoulos, and I can also arrange this as a time series, as a temporal sequence like in this case. Now, I could use this to reconstruct a trajectory that could come out, right. So, this is the population activity when the movement is A, or when I am going from outside to the inside, when the spiral is spiraling in, right. So, this is the activity of the neurons, this is population activity. Likewise, when you are going out from in, I am recording activity, and that is also represented as instantaneous directions and velocities vectors, right. From these vectors it is possible for me to reconstruct trajectories, just like it is possible for me to decompose these trajectories into these, or more precisely this was B is decomposed into the vector C and A is decomposed into this vector A, is it not? Just like I have decomposed the movement trajectories into these vectors, I can also use these vectors to reconstruct trajectories theoretically, and that was attempted. When that was attempted, this movement activity, this activity of the neurons results in a trajectory such as this one, and D results in a trajectory such as this one. Now, let us take a look and see what happens. So, what was recorded? Actual movement trajectory was recorded and activity of 240 neurons in the primary motor cortex was recorded. From the activity of the 240 primary motor cortex neurons, we are able to get the instantaneous directions and velocity vectors, from which we are able to reconstruct the preferred, the approximate trajectories. Those are given in C and D. So, this is the actual movement, this is the prediction. Okay. C and D are predictions, A and B are actual trajectories. Compare A and C, for example, you will find that C is not an exact reproduction of A, yet there is a striking resemblance, is it not? So, I am not able to exactly reconstruct as it is in A, because A looks smooth and much more neat, but the reconstruction from neuronal activity is very similar. It is not the same, but it is very similar. Likewise for B and D, right. B and D, if I compare, D is not an exact reproduction of B, but it resembles very closely, or at least there are many features that are very similar. So, from this what we can conclude is that activity of a population of neurons predicts not just the force that needs to be produced, not just the movement that but also the instantaneous directions and velocities. So, it predicts kinematics over time. The activity of neurons precedes the actual trajectories by about 100 milliseconds. Okay. And note one more thing, if I had units for these trajectories, say this is the y axis, this is the x axis, and in centimeters this is x in centimeters, this is y in centimeters, I am not going to be able to get these trajectories also in in centimeters, So, in other words, I am going to get characteristics that are similar, but not exact magnitudes. So, the characteristics are going to be strikingly similar, but the exact magnitudes and units will not be similar, because why? From velocities I am decomposing into displacements, there are going to be some errors that are going to arrive. So, I am not going to be able to exactly predict this moved by so many centimeters, but I can predict the approximate trajectory of movement. So, the characteristic of movement I can predict. Okay. So, this is the work of Andy Schwartz. So, this is Moran and Schwartz 1999, thanks to our colleagues for sharing this data. Okay. So, next we discuss what happens when the monkey is trying to move against external loads. So, I said earlier in the Georgopoulos experiment there was no load, right. The monkey is trying to make movements to the 8 directions, no load. Suppose the monkey is making a movement and suddenly an external load is either assisting the movement or opposing the movement in a certain different direction. What could happen? That is the question. So, here, and what was found was that depending on the direction of force that is required, right. So, depending on the load that is acting, the direction of force that is required will change. Depending on the direction of force that is required, the activity of the neurons does change. For example, here what is presented is the no load activity. Okay. So, when there is no load, if the movement is in a particular direction. So, this is the original position, this is the home position. From this point the monkey is required to move to that direction. The position of each of these group of activity represents the position that was required to be moved. Okay. So, that is the position that was required to be moved. For example, that was the target, and the activity of these neurons predicts that direction. Likewise, this was the target and the activity of these neurons predicts that direction, for example, and so on and so forth. Look, this is approximately a repetition of or a replication of the Georgopoulos experiment. But what is new in this experiment is what happens when the monkey is required to move against a load. Now, the load in this picture in B, the position, the direction in which the load was pulling is what is represented here. So, this shows the direction in which it was pulling, okay, not the target to be moved. This is the direction in which it was pulling, and I have to. So, in that case then the monkey is or its neurons are acting in the opposite direction. Likewise, the load is pulling in that direction, is it not? So, that is the center, the load is pulling in that direction, the monkey's neurons are acting in the opposite directions. So, once again showing that depending on the external load, if I have to reach to a particular target or if I have to maintain the direction of a movement or then depending on the direction of the load the activity will change, and it does change. Okay. So, this is again data from Kalaska, right. Once again we are thankful to our colleagues for this. Now, what about this is again activity of a population of neurons, right. So, that was also the case with the previous Andy Schwartz and his group. What happens with individual neurons, right. So, this is data from Sergio and Kalaska, right. So, here what is presented is there is not an actual movement. This is the case of isometric force production, or you have a manipulandum, you have an object that is immovable, that is strongly fixed to a table, and the monkey is given a cue as to which direction in which it is supposed to produce a force. It is trying to push this object in a particular direction, but it cannot push it because the object is fixed strongly. Let us suppose that is the case here. Now, I can push it in that direction, I can push it in that direction, I can push it in many different directions, is it not? Now, this is activity from this picture presents activity from a single neuron. So, what about this case? What about the case of Kalaska? This is activity from about 260 M1 neurons. Okay. This picture, whereas this picture presents activity from a single neuron, and when the monkey is producing force in a particular direction, there is maximal activity as seen by these, and in the opposite direction there is very minimal activity, did not. So, from this I can again reconstruct a directional tuning curve like in the case of Georgopoulos, and that is shown here. Now, let us discuss the fundamental differences between the Georgopoulos experiment and this experiment. In this experiment there is not an actual movement. In the Georgopoulos experiment, so there was a home position and there was a target, there was actual reaching, there was a change in kinematics. In this case there is no change in kinematics, the monkey is holding the object at the same position throughout the trial, yet the activity change is representative of the direction in which it is pushing the object. So, the direction of the isometric force that is being produced is also represented in a way that is somewhat similar to the direction of actual movements, or in other words this is kinetics, is it not? This is kinetics. Why? This is actual force production, whereas Georgopoulos experiment is reaching or generation of trajectories or kinematics. So, it seems that the neurons behave in approximately similar ways in both cases. Okay. So, this is data from Sergio and Kalaska, alright. So, from this what we have seen from these studies is that from the work of Andy Schwartz, we can say that depending on the population activity of a bunch of neurons, we could predict the actual trajectories, we could reconstruct the trajectories to some approximation. In other words, we can reconstruct the population vectors, we can reconstruct the direction and instantaneous velocities, and then using that we could reconstruct the displacement trajectories, which will be approximately similar to the actual movement trajectories. This is the work of Andy Schwartz. Then the work of Kalaska, where depending on the external load direction, the activity of a population of neurons will vary, and depending on the isometric force production direction. So, even if there is no real movement, the direction of force production, the direction in which the force is produced on an object can be encoded or is preceded by activity of individual neurons in the primary motor cortex. So, with this we come to the end of this class. We will continue the lecture in future classes. Thank you.